Begin by cloning the MedGT public repository and follow the setup instructions to launch the application. Once MedGT is running, access the platform and create a new annotation project. Configure the label schema by selecting from the available predefined clinical labels or by creating custom labels based on the study requirements. If a DICOM study is not available, download the sample MRI study from the public storage bucket. Upload the DICOM data to the project. MedGT processes the study and prepares it for visualization and annotation. Role-based access ensures that each user can perform only the actions relevant to their assigned responsibilities. Users can be configured as annotators, clinical reviewers, or administrators. Once the users are added, assign the required annotators and validators to the study. Open the study in the medical imaging viewer and switch between axial, sagittal, and coronal views. Use zoom and pan controls to inspect anatomical details and navigate the image. Enable MPR for synchronized review across multiple imaging planes. Enable the crosshair tool to localize the same anatomical point across all synchronized views. Moving the crosshair in one plane automatically updates the corresponding position in the other views. Select the MedSam tool and choose the required clinical label. Draw a bounding box around the target anatomical structure. MedSam automatically generates the segmentation mask within the selected region. Use interpolation to automatically generate annotations across the intermediate slices. Once the annotations are complete, submit the study for review. The submitted study enters a multi-validator clinical review workflow. Each clinical validator can independently review the study and perform annotation-level quality assessment. Individual annotations can be marked as accepted or rejected. When an issue is identified, the validator can precisely localize it by drawing a review box around the affected region. A review comment can also be added to describe the issue, such as over-segmentation, under-segmentation, incorrect boundary placement, or missed anatomy. A study rejected by Clinical Validator 1 remains available for independent review by Clinical Validator 2. All review decisions and comments are retained as part of the clinical validation audit trail. When changes are requested, the study status is automatically updated to changes requested. The annotator can review the validator comments and navigate directly to the marked issue. The identified annotation can then be corrected based on the clinical feedback. After completing the corrections, the annotator resubmits the study for review. The study status is automatically updated to in review. The clinical validator reviews the corrected annotations and confirms whether all requested changes have been addressed. Once the task passes clinical validation, the validator approves the study. The study status is then automatically updated to approved. There are two export options, JSON and DICOM seg. Export the study as DICOM segmentation, preserving the segmentation masks together with their associated anatomical labels and source image references. The exported study can then be loaded into a compatible DICOM viewer to overlay the segmentations on the original images for clinical visualization, verification, and review. Approved annotations can also be exported in JSON format to view the annotation data and metadata.